Very good morning to all the CUET aspirants. So here we are in our third session of Microbial Diversity, where we are going to learn in detail about archaea, eubacteria and diseases that are caused by phytoplasma and spiroplasmas. And we are also going to address actinomycetes. Fine, right? Let's get started without much delay. So this is the basic introduction of archaea. Archaea are usually called as what? Common ancestors, or the ancestral group of prokaryotes, because in Greek archaeos means what? Ancient. Okay, and they show wide range of morphological and physiological diversity. They are distinct from bacteria and eukaryotes. This distinct nature is shown in 3 domain and 6 kingdom system of classification by Carvus. Okay, Carvus has given 3 domain and 6 kingdom system of classification, where initially he has said that there is a common ancestor called as progenote. Progenote is the common ancestor because it has 16 srRNA, which is common in all the 3 domains. And what are the 3 domains for which progenote is the common ancestor? 3 domains include archaea, bacteria and eukarya. Fine, right? And 6 kingdoms domain. Under archaea domain we have archaebacteria, under bacterial domain we have eubacteria, under eukarya we have 4 kingdoms. Fine, right? What are they? Protista, fungi, plantae and animalia. Fine, right? So this is the basic classification that we have to know. How come archaea is differentiated from bacteria? 2 main reasons. One is its cell wall composition and the second is its the split gene arrangement of mRNA. Okay, archaea is having split gene arrangement, that is intron exon sequences, like how you will see in the eukaryotic cell. In bacteria you don't have such arrangement. This is addressed as reminiscence of antiquity. Okay, what is it, ma? Reminiscence of antiquity. Intron exon sequences is nothing but reminiscence of antiquity, which is reminding about the ancestral cell. And second most important feature is the cell wall. Cell wall of archaea is composed of lipopolysaccharides rather than proteins. Okay, rather than amino acids, it is widely composed of lipopolysaccharides. That is the main reason where archaebacteria are adapted to diverse environmental conditions. It can be thermoacidophils, methanogens or halophiles. Clear, right? Now when you look into the morphological diversity, they can be as spherical, rod shaped, spiral, plate like lobed or irregular. Pleomorphic we will say it as. Pleomorphic means which change their morphology very often. And we can have gram positive and gram negative bacteria, archaebacteria. Organization, if you see, they can be unicellular or aggregated into filaments. We have 0.1 to 15 micrometers to 200 micrometers length of archaebacteria, that much diameter. When you look into the physiology, we have diversity in respiration and nutrition. Respiration diversity, if you see, they can be as obligate anaerobes, facultative anaerobes and aerobes. If you look into nutrition, they can range from chemolithoautotrophs to organotrophs. They can range from chemolithoautotrophs to organotrophs. Clear till here, ma? And then if you observe reproduction, reproduction can be for unicellular archaeal forms, it can be through binary fission. For multicellular or colonial forms, they can be budding, through budding and fragmentation. They are often found in extreme environments like hyperthermophiles, mesophils, hypersaline and cold environments as well. These are the extreme environments where archaebacteria are present. And some live as symbionts in animal digestive system like in ruminants. In ruminants, archaebacteria are present. In the gut of ruminants, archaebacteria are present, wherein these are addressed as methanogens. Methanogens. Fine, right? Archaea may make up 34% of prokaryotes in coastal Antarctic waters. Very important point to be highlighted and noted. 34% of prokaryotes are archaea only in coastal Antarctic waters. When you look into the cell wall composition of archaea, we have two types of cell wall. One is gram positive and one is gram negative. Gram positive bacteria, here you can see in the cell wall there is high amount of lipoproteins and peptidoglycans. Thick homogeneous outer layer is present, and this thick homogeneous outer layer is composed of lipoproteins and peptidoglycans, which will retain the stain, which will absorb the stain strongly. Whereas in gram negative bacteria, they lack outer membrane and complex peptidoglycan is absent. They do not have complex peptidoglycan and the outer membrane is absent in gram negative archaea. Till here it is clear, right? Yeah, cell wall composition is clear to us, and in what environment they can survive is also clear to us. Now, what are the key differences of archaea from bacteria? We will highlight. Archaea lack muramic acid and amino acids, as I told you. D amino acids and muramic acid is absent in the cell wall of archaea. That is the major point. Second is archaea resist the attack by lysozyme, which is released by viral particles or bacteriophages. Lysozyme will degrade the cell wall of archaebacteria. Archaea cannot resist, archaea will resist the attack by lysozyme and beta-lactam antibiotics, which are not present in bacteria. For example, penicillin. Okay, archaea will resist the attack. Next examples, if you see, Methanobacterium can have pseudomurin in its cell wall. They have L amino acids, D amino acids are absent. Cross linking is by N-acetylosamuramic acid. Okay, N-acetyl-talosamuramic acid. Fine, right? Now, what type of bondings are included in this lipopolysaccharides? Beta-1,3 glycosidic bonds. Usually we have beta-1,4 glycosidic bonds in between the lipoproteins or peptidoglycan which is present in the cell wall. But instead of beta-1,4 glycosidic bonds, here we have beta-1,3 glycosidic bonds. Fine, right? And cell wall composition in archaea, Methanosarcina and Halococcus. Okay, in Methanosarcina and Halococcus, they contain complex polysaccharides similar to that of chondroitin sulfate. Instead of pseudomurin, they have complex polysaccharides which are similar to chondroitin sulfate. Fine, right? Now, gram negative archaea, they have a layer of protein and glycoprotein. Okay, they have a single layer of protein and glycoprotein. Outer membrane is absent. Cell membranes are single and double layered, which are 20 to 40 nanometers thick. Fine, right? And wall composition will vary by genus, like methanogens or methanolobus in glycoprotein walls, sulfolobus where protein and in glycoprotein walls, methanococcus and methanobacterium have protein walls, and desulfurococcus, they also have protein walls. Here what you have to remember, with the example you just highlight they only have protein walls or they also have glycoprotein walls, that is it. Fine, right, ma? So, this is the complete description about the key differences from bacteria, very important, and cell wall composition in archaea. And then if we move on to membrane lipids, what type of lipids are present in archaea? Most distinct feature is their membrane lipids. They have branched chain hydrocarbons. Branched chain hydrocarbons with what linkages? Either linkages. In bacteria you can see ester linkages in bacteria as well as in eukaryotic cells. If you observe, be it in single celled eukaryotes like protists, you will have ester linkages, but here we do have either linkages, that are branched chain hydrocarbons. They have branched chain hydrocarbons linked to glycerol by what? Either linkages. Next, polar lipids will have phospholipids, sulfolipids, glycolipids. Fine, right? Membranes may have diethers, tetraethers and monolayers with extreme stability. Sulfolobus, they have almost completely tetraether monolayers. Okay, they have tetraether monolayers. Clear till here? Any doubt anywhere, ma? Yeah. Now, genetic material and ribosomes. Archaea chromosomes, they have single closed circular chromosomes. single double stranded circular chromosomes are present, single stranded, and they have smaller genomes when compared to bacteria. For example, E. coli, whose molecular weight of nucleotides will be 2.5 into 10 to the power of 9 Daltons. Thermoplasma acidophilum, they have 0.8 to 10 to the power of 9 Daltons. They show high variation in guanine plus cytosine content. G plus C content is high in the nucleotides or in the genome of archaebacteria. And mRNA is similar to bacteria, there is no splicing. Okay. Promoters, sorry, this statement is wrong. mRNA is similar to eukaryotes, it is not similar to bacteria. Splicing occurs. And promoters resemble bacterial ones. Ribosome is of 70S type, whose function is similar to the eukaryotic ribosomes. For example, the ribosomes here in archaebacteria is sensitive to anisomycin and it is resistant to chloramphenicol and kanamycin. These drugs you have to remember for which it is sensitive and for which it is resistant. Clear till here, ma? Yeah. We will move on to the reproduction in archaea. Here we go with reproduction and classification of archaebacteria. Based on the ribosomal RNA analysis, second edition of Burgess manual has classified archaea into two categories. One is Euryarchaeota and Crinaarchaeota. These are the two classifications. Euryarchaeota, both are derived from Greek only. This means wide ancient archaebacteria. This means spring ancient archaebacteria, Crinaarchaeota. Latest Burgess manual divides archaea into 5 groups. One is methanogenic archaea, second extreme halophiles, cell wall less archaea, extreme thermophilic sulfur metabolizers and sulfate reducers. This is the latest division of archaea into 5 groups. The basic division we know into thermoacidophils, halophils and methanogens, but this is the advanced classification. Clear till here, ma? Yeah. Next we will move on to general characteristics of actinomycetes. They morphologically resemble fungi. Same like archaebacteria, they also possess high guanine cytosine content. They also possess high guanine cytosine gram positive filamentous bacteria. Okay, and they adapt to solid agar medium. They adapt to solid agar medium by forming branching hyphal networks. They form branching hyphal networks. They grow on substrate surface and they penetrate inside. They form strong substrate mycelium as well. Septa will divide the hyphae into multinucleated cells. Okay, the filamentous algae where septa will divide this into cenocytic condition or multinucleated cells. Cenocytic condition or multinucleated cells like this. Septa divide hyphae into multinucleated cells. Aerial mycelium at hyphal tips. They form conidiospores which aid in asexual reproduction. Okay, conidiospores, remember that is a specific feature of actinomycetes. And sporangiospores, they form what? They are formed in sac-like structures. Sporangiospores are formed in sac-like structures. Spores, both conidiospores might be, sporangiospores might be, they are resistant to desiccation and not heat resistant. They are not heat resistant. Reproduction is by binary fission, budding and fragmentation. Okay, reproduction is by binary fission, budding and fragmentation. These are the general characteristic features of actinomycetes. Now we will move on to cell wall composition. Classification of cell wall, this is based on peptidoglycan composition. Okay, cell wall classification of actinomycetes, this is also based on peptidoglycan composition. We have 4 cell wall types here. Main criteria if you see, amino acid in tetrapeptide side chain. Okay, tetrapeptide side chain amino acid, this is put in position 2. Presence of glycine in interpeptide bridges. Third point is peptidoglycan sugar content. This is important, fine, right? These are the main criterias for classification of actinomycetes based on the different cell wall types. What are the criterias we are considering, ma? One is amino acid in the tetrapeptide side chain. Second, presence of glycine in interpeptide bridges. Third is peptidoglycan sugar content, fine, right? Now, examples if you observe, nocardia and Saccharomyces, okay, this is one example. They contain arabinose and glucose and no glycine, they do not have glycine. These are kept under type 4 cell wall, okay. Streptomyces land under type 1, Monospora under type 2, actinoplanes and Frankia is under type 3. So, you are clear with 4 types, type 1, 2, 3 and 4, clear with this? Now, ecological and practical importance. First is free living soil bacteria, and they act as saprophytes, degrading organic matter. They are important in mineralization of soil organic compounds. They produce wide range of natural antibiotics, and some are pathogenic to humans, animals and plants, okay. So, these are of ecological and practical importance. Clear till here? Any doubt anywhere, ma? What are the features of some genera of actinomycetes? We have 2 such genera. One is actinoplanes and second is Arthobacter. Morphology if you observe, these are filamentous hyphae where sporangia are formed, motile spores are present, and these are of polar flagella. Guanine cytosine content is 72 to 73% in their nucleotides or in their genome. And what type of cell wall type here it is? This is aerobic soil microbes kept under cell wall type 2. The types of actinomycetes is very important, okay, remember that. Next is Arthobacter. These are non-motile cocci or rods. Guanine cytosine content is 59 to 70% approximate. They are aerobic and soil microbes only. And then here we go. Actinomycetes, they are mostly aerobic and free living. Pathogenic genera of actinomycetes if you observe, Mycococcus and Mycobacterium. Frankia will form nodules in Casuarina and other non-legumes. They fix nitrogen inside the globular cyst. Okay, inside a globular cyst the nitrogen is fixed like rhizobia, Next, Streptomyces, they produce antibiotics. For example, Streptomyces griseus, they produce antibiotic, that is streptomycin, and Streptomyces cabeus, they cause what disease? Cabeus disease in potatoes. Points to be noted and highlighted, ma, just go through. Next, these are different genera again. What is the most important points to be noted over here? Under each example, please look at the type of the cell wall to which it belongs to, guanine cytosine content, and what is the basic morphology of that. If you are having any doubts, you can let me know. You see the genus Frankia, Micrococcus, Mycobacterium, Nocardia and Streptomyces, fine, right? What type of cell wall it is having, under what type it is fit into, and what is the guanine cytosine content percentage is present, morphology of that to be noted, fine, right? Any doubts anywhere, ma? Yeah, now we will move on to Mycoplasma, which is very important, okay? Mycoplasma, which are addressed as jokers of plant kingdom because they They are pleomorphic in nature, they change their shape very often and they are wall-less bacteria. They are also addressed as bacteria with their coats off. Okay, they do not have cell wall. Mycoplasmas are the smallest wall-less free living prokaryotes. They belong to molecules soft skin of schizomycetes. They are discovered by Pasteur in the year 1843 during their studies on pleuropneumonia. Initially they are called as pleuropneumonia-like organisms. Okay. The name Mycoplasma was coined from Greek. Mycos means fungus, plasma which is formed. First used, this is Mycoplasma which is first used by Albert Bernhard Frank, and they are isolated by Nocard and Rox in the year 1898 from cattle pleural fluids. They are isolated from pleural fluids. Discovery of Mycoplasma like organisms. Okay, how they are discovered? Mycoplasma like organisms. In 1967, Doi, Ishii and co-workers, they are discovered cell wall-less microbes in phloem tissue of yellow diseased plants. They named them as Mycoplasma like organisms or Mollicutes like organisms. They are confined only to the phloem and they are lagged cell walls. They do not have cell walls. Fine, right? See here you can see the structure clearly. Mycoplasma without any cell wall and DNA double stranded linear or circular. They have soluble proteins and they have 3 layered cell membranes. Soluble RNA is also present here and ribosomes which are of 70S type. Fine, right? This is the cellular composition of Mycoplasma. Clear, right, ma? Now, morphology and movement. Of course, they are pleomorphic. Mode of existence is mode of nutrition also you can say. They exist as parasites or saprophytes. Spiral shaped forms. Okay, spiral shaped forms of Mycoplasma, they are kept under spiroplasmas. Elongated shaped are kept as phytoplasma, spiroplasma and phytoplasma. Usually they are non-motile, some show gliding movement. Okay, some Mycoplasma they show gliding movement. Taxonomic placements, if you see, they are belong to single class with how many orders? 5 orders and 6 families. Previously they are grouped into single order, those are Mycoplasmataceae. Families include Mycoplasmataceae, Spiroplasmataceae, Acoleptoplasmataceae, Asteroplasmataceae, Eurycyphoplasmataceae. All these are not included, not required for you, just remember the examples. Most importantly, Spiroplasmas and Phytoplasmas. These orders and all not required for you. Morphology of Mycoplasma. They are pleomorphic. Mycoplasmas can change their shape which depends on growth conditions and medium and host cell. There is no cell wall for Mycoplasma. They cannot synthesize peptidoglycan precursors. Okay, they do not synthesize peptidoglycan precursors which is surrounded by cytoplasmic membrane, and I told you they are surrounded by 3 layers of cell membrane which are lipoproteins in 3 layers. Okay, these are lipoproteins in 3 layers. Shape and size, spherical and pear shape 0.3 to 0.8 micrometers, helical branched filamentous forms, colonies on agar which resembles like fried egg appearance. Okay. What are the key features? They are distinct from bacteria and plant viruses. Comparison or comparative analysis, if you see, although they resemble as mutated bacteria with lost cell walls, Mycoplasma will never regain the cell wall because they are wall-less. They resemble protoplasts but are osmotically stable. Okay, Mycoplasmas are osmotically stable. Stability is due to what? Sterols in their plasma membrane. Very important point to be noted. Okay, why there is stability for Mycoplasmas? That is because of the sterols which are present in the cell membrane. Fine, right? Membranes and surface components. Some require sterols for growth, some will have lipoglycans. They are similar to lipopolysaccharides in gram-negative bacteria. They aid in attachment to the host cell. They help to stimulate antibody production. They help in stimulating antibody production. Phylogenetic status, Mycoplasma has no history of reversion or derivation from typical bacterial forms. Considered intermediate organisms between bacteria and viruses. Fine, right? They are considered as intermediate organisms between bacteria and viruses. Clear, right? These are the key features of Mycoplasma. What is the cytoplasmic content and what will be its antibiotic sensitivity? Antibiotic sensitivity is always major point to be highlighted all the time. Here we go with cytoplasmic content. They contains double-stranded DNA, RNA, ribosomes and proteins which are dissolved. Nucleic acid ratio, DNA is of 4% and RNA 8% in the complete cytoplasm. Spherical bodies, they are found in cytoplasm which are of 60 to 100 nanometers in diameter. Here what is more important is the percentage of DNA and RNA, nucleic acid ratio in the cytoplasm. They form structural variations, mycoplasmas in phloem which are spherical, aster yellow mycoplasmas, they lack filaments. They appear as spherical to oval shape. Budding or binary fission, they are generally not observed. Budding or binary fission is not observed. Next, antibiotic sensitivity. Ineffective antibiotics target cell wall. What is the target cell wall? Which targets cell wall? Ineffective antibiotics like penicillin, ampicillin, methicillin. These are not sensitive by Mycoplasma because Mycoplasma does not have cell wall and these antibiotics will target the cell wall. So, they do not show any effect on the Mycoplasma. If you see effective antibiotics which acts or targets on the metabolism of Mycoplasma, they show huge effect in the functioning of the Mycoplasma. What are those effective antibiotics for Mycoplasma? Oxytetracycline, streptomycin, erythromycin, chloramphenicol. These are the effective antibiotics on Mycoplasma. Remember that. Next, nutritional and culturing characteristics. They can be used as saprophytes or parasites. They grow well on artificial medium and parasitic forms they grow on medium with protein, sterol, phospholipids, mucins, DNA and RNA. Reproduction is by budding and binary fission. Fine, right? What are the unique features? They vary small genome size, simple structure, they do not show any gram staining. Unlike actinomycetes or eubacteria which you have seen, archibacteria which you have seen previously, Mycoplasmas they do not show gram staining, gram positive because they do not have cell wall. Related to low GC gram positive bacteria. Previously actinomycetes or archibacteria they are having high guanine cytosine content, but here you have low guanine cytosine content. They can pass through filters that normally block the bacteria, okay. They can easily pass through bacterial filter paper, bacterial filter paper. Fine, right? They can easily pass through bacterial filter paper. Now, diseases caused by Mycoplasmas. There are so many diseases which are given. Just go through the concept, go through the diseases that are being caused by Mycoplasma, but we have to learn few diseases in detail. Just go through the diseases, like many species of Mycoplasma or Mycoplasma like agents, they cause serious diseases in human beings. Like Mycoplasma will affect the respiratory organs, circulatory system, cardiovascular system and urinogenital tract as well. The agent which causes primary atypical pneumonia PAP is Mycoplasma pneumonia disease, okay. Mycoplasma pneumonia. Symptoms include pneumonia, hemorrhagic laryngitis and vesicular inflammation of tympanic membrane, okay. Next, other mycoplasmal species like Mycoplasma humanus, they cause what? Pleuropneumonia, inflammation of genitals and non-specific urethritis, prostatitis, endocarditis and other conditions, infertility as well. Mycoplasma humanus and Mycoplasma fermentatus are known to cause infertility in man. Okay, these are the diseases that are caused by Mycoplasma. Till here everything is fine, no? The diseases are very important. Just go through that and note it now. Now, in human beings, Mycoplasma incognitus. Recently, this is discovered in spleen, liver, blood of AIDS patients, which causes infections. What are the common pathogens in humans? Just take a note of it. And in animals, you see, Mycoplasma will cause diseases in sheep, goat, dogs, rats and mice. They can be like rheumatic arthritic diseases, respiratory tract infections and infections in mammary glands. Okay. In plants, if you observe Doi et al. And Ishii et al., they have discovered Mycoplasma like organisms or molecules like organisms which causes witch's broom disease and yellow type diseases. Fine, right? Initial believed to be viral diseases like Marmorsh, they are confirmed Mycoplasma like origin. Fine, right? These organisms are now termed as phytoplasma. Okay, they are now addressed as phytoplasma. Clear, right? Clear till here? Any doubt anywhere in both plants and animals? Now, we will see both the types, phytoplasmas and spiroplasmas. First, what is the characteristics features of phytoplasma? They are wall-less prokaryotes belonging to molecules. Obligate parasites, they lack cell wall, we know that, pleomorphic or filamentous type of Mycoplasma. They are discovered in ultra thin sections of phloem showing yellowing symptoms. What are the symptoms that are caused by phytoplasma? It can be yellowing, phylloidy, dwarfing, ination and witch's broom. They affect crops like rice, maize, brinjal, cotton, citrus and potato. Phytoplasma are known to cause 75 types of plant diseases in 300 plus plant species. Clear? Important diseases by phytoplasma. We have like clover dwarf disease, maize dwarf disease, aster yellow, bunchy top of papaya, rice stripe, yellow dwarf disease, witch's broom, peach yellow, clover phylloidy. Coconut lethal yellow, yellow dwarf of pear, tobacco, sesame phylloidy, witch's broom of groundnut, white leaf disease of sugarcane, sandal spike disease, little leaf brinjal, witch's broom of alfalfa, gracy shoot sugarcane, sunflower phylloidy. These are the diseases that are caused by phytoplasma. Clear? Different diseases caused by phytoplasma. Any doubts anywhere? Yeah, and then now we will see the type studies of little leaf brinjal. In each type study, what you have to highlight is the symptoms. What are the symptoms that are caused and what is the disease? What is the importance of this? Fine, right? So, the disease is little leaf brinjal, host is Solanum melongena, which is commonly addressed as eggplant or brinjal. Molecules like organisms like phytoplasma will cause the disease. Importance, if you see, one of the major destructive diseases which causes huge economic loss. Marked symptoms, which are the major symptoms of this disease in little leaf of brinjal, is marked reduction in the size of leaves. Size of leaves will be reduced, which is characteristic symptom of this. Symptoms of little leaf disease of brinjal, if you see, they are newly formed leaves. They become progressively smaller and sterile. Petioles are very much shortened. Leaves are appressed to the stem. They become narrow, soft, glabrous, yellow. Phylloidy and proliferation of axillary buds. Internodes are shortened. Lamina, which is leaf lamina, they become soft and glabrous. Top branches show shortened internodes, and late infection leads to small and hard fruits. These are the major symptoms. Just take a note of it, ma. What is the pathogen that is causing? Phytoplasma or mycoplasma like organism, they have a wide host range. They can cause disease in brinjal, tomato, potato, dadura and ashwagandha. Transmission is by vectors like leaf hoppers. What are the vectors of this disease is also important. Fine, right? Hishimonas phycetes, Emphosia devastena, these are reported by Thomas and Krishnaswamy. Reporting you leave, but what are the vectors that can cause the disease, that can transmit the disease is important. Control measures can be field sanitation and timely planning or timely planning of plantation is important. These are the control measures. Next is papaya leaf curl disease. Here also what is important? Symptoms are important. And what is that is causing here? Host is Carcina papaya and molecules like organism, phytoplasma is causing the disease. They are affecting young leaves. Top leaves show curling, crinking and distortion. Petiole twisting happens, reduction of leaf lamina. Leaves roll downwards and inwards. Thickening of veins. Leaves become lethargy, brittle and distorted. No flowering or fruit production is seen. Advanced stages causes defoliation also and the growth is arrested completely. Pathogen, molecules like organism which has single stranded DNA, not seed or soil borne, they are not sap transmissible and transmission is via grafting. Vector is Bemisia tobacci, okay, which is silver leaf whitefly. This is the vector of this disease. Control measures you can take down. Most time repeatedly asked questions is from symptoms and control measures like what type of sprays, foliar sprays we are using, okay. Next is spiroplasmas. Spiroplasmas, unlike phytoplasmas, they are helical, wall-less bacteria, same wall-less bacteria, but they are helical. They are pleomorphic again, spiral or helical shape. Thus, because of its shape only we are calling it as spiroplasmas. Morphological features, shape is helical, flexible or pleomorphic, width is 0.2 to 0.3 micrometers, length is 2 to 8 micrometers. Cell wall is completely absent. It has a single 3-layered membrane and corkscrew like movement is shown which is using internal cytoskeleton. Filaments, no flagella is present over here. Genome is small circular double stranded DNA. They range from 0.1 to 0.4 micrometers. Replication is majorly by binary fission. Here we see what type of diseases are caused by spiroplasmas and what are the vectors. The single table and the symptoms are enough from this slide. Symptoms that are caused by spiroplasma and what are the diseases. Constant disease, citrus stubborn disease, periwinkle yellow disease, honeybee, May disease. These are the major diseases that are caused by what? Spiroplasmas, and these are the vectors through which they are transmitted. Next, control measures. These differences are important. Fine, right? Last we will end up with the differences of spiroplasma and mycoplasma. Both belong to the bacteria only. Class is Mollicules. Cell wall is absent. Fine, right? Spiroplasma are helical shape, phytoplasma are pleomorphic. Usually they are spherical or filamentous. Motile via corkscrew motion, and phytoplasmas are non-motile. Genetic material is double-stranded DNA, and here these are also double-stranded DNA. Genome size is 1.0 to 1.4 micrometers, and here 0.6 to 1.0. Spiroplasmas shows their effect both in plants and insects, but phytoplasma will infect only plants. Transmission is via insect vectors. This is via phloem feeding insects. Okay, phytoplasma, this is via phloem feeding insects. Major plant diseases include constant citrus stubborn disease, phytoplasmas, little leaf of brinjal, grassy shoot of sugarcane, and aster of yellows. These are the diseases of phytoplasma, major plant diseases. Next you observe spiroplasma and phytoplasma. Few more differences. Tissue infected in plants, both affect phloem only. Symptoms: stunting, yellowing and bushy plant. Here phylloidy, yellowing and witch's broom and sterility. These are the symptoms. In vitro culturing, they can be cultured in artificial media in sp4 broth. They cannot be cultured in vitro as they are obligate parasites. Detection is by electron microscope and PCR, same ways. Known vectors are leaf hoppers, aphids and beetles. Here in phytoplasma, leaf hoppers and plant hoppers. Two diseases, their major symptomology is important. Leaf curl in papaya and little leaf brinjal. Both the diseases are important, they are symptoms. Key differences between spiroplasma and phytoplasma. Fine, right? So, all the points which are discussed in the class are important for your exam. Just go through them. If you are having any doubts, drop a message in the comment box. I will be addressing your doubts. And second point is, in our next class, we are winding up the microbial diversity with objective practice session. So, come prepared with a complete microbial diversity. We will have a very good objective session. So, that is all for this session. Thank you.